Hello everyone, welcome to Nazia Forum. Today ನಾನು ನಿಮಗೆ 9th ಸ್ಟ್ಯಾಂಡರ್ಡ್ದು ಸೈನ್ಸ್ ಸಬ್ಜೆಕ್ಟನ್ನು ಇಲ್ಲಿ ಇಂಟ್ರೊಡ್ಯೂಸ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. 9th ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಸೈನ್ಸು. ಓಕೆ, ಒನ್ ಬೈ ಒನ್ ನೋಡೋಣ. ಇಷ್ಟವಾದರೆ ದಯಮಾಡಿ ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ, ಲೈಕ್ ಮಾಡಿ, ನಿಮ್ಮ ಫ್ರೆಂಡ್ಸ್ ಜೊತೆ ಶೇರ್ ಮಾಡಿ. ನಾನು ಇಲ್ಲಿ ಡಿಸ್ಕಸ್ ಮಾಡುವುದು ಅದು HSTR ಇರ್ಬೋದು, GPSTR ಇರ್ಬೋದು, ಅಥವಾ PSTR ಇರ್ಬೋದು, ಅಥವಾ 9th ಸ್ಟ್ಯಾಂಡರ್ಡ್ ಕಲಿಯುವಂಥ ಮಕ್ಕಳು ಇರ್ಬೋದು, ಎಲ್ಲರಿಗೂ ಯೂಸ್ಫುಲ್ ಆಗ್ತದೆ. ಸೊ ಇದನ್ನು ಕೇರ್ಫುಲ್ಲಾಗಿ ನೋಡಿ, ವಿತ್ ಎಕ್ಸ್ಪ್ಲನೇಷನ್ ನಾನು ಆನ್ಸರ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. ಸೊ ಕೇರ್ಫುಲ್ಲಾಗಿ ನೋಡಿ, ಪೂರ್ತಿಯಾಗಿ ವಿಡಿಯೋವನ್ನು ನೋಡಿ, ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ. ಧನ್ಯವಾದಗಳು. ಓಕೆ, ನಾನು ಸ್ಟಾರ್ಟ್ ಮಾಡ್ತಾ ಇದ್ದೀನಿ. Okay, important question for class 9th science, that is chapter number 5, the fundamental unit of life. Can you name the two organelles we have studied that contain their own genetic material? Okay, what they are asking here, the organelles which contain their own genetic material. What is genetic material? Genetic material means the organelle which has contained own DNA and RNA. Own DNA and RNA. They both are the genetic material. DNA and RNA are the genetic material. The organelles which has contained own DNA and RNA, that is we call it as a such organelles are called own genetic material organelles. Okay, the examples are chloroplast and mitochondria. They both are the example for this own genetic material organelles. Okay, next come to second question. What are the protein synthesis inside the cell? Protein synthesis, a protein house of synthesis takes place in the ribosome. Very important. Where protein takes place, synthesis takes place in ribosome. Ribosome is production of protein factory. In this ribosome are the place where protein is synthesized. Inside the cell, ribosomes are present. In that ribosome, proteins are produced. Okay, next come to next question. Who discovered the cells and how? Robert Hooke discovered the cell. He is an English scientist. Discovered cell in 1665 while examining the tiny slice of cork under his self-designed microscope. He saw honeycomb-like structure. Okay, he saw honeycomb-like structure. Who discovered the cell? Robert Hooke. He is a British or an English scientist. Why is the cell is called structural and functional unit of life? That is very important question. Why cell is called structural and functional unit of life? Because all living organisms are made up of cells. Every living organism is made with a cell. The cell is the basic construction unit of living organism. It is a basic construction of living organism, and all of the living organism activities are the sum of activities are conducted by its cell. The cell is referred to as an structural and functional unit of life. ಯಾವ ರೀತಿ ಅಂತಂದರೆ, ಒಂದು ಮನೆ ಕಟ್ಟಬೇಕಾದ್ರೆ ಯಾವ ರೀತಿ ಇಟ್ಟಂಗಿಗಳು ಒಂದಕ್ಕೊಂದು ಇಟ್ಟು ಮನೆಯನ್ನು ಕಟ್ತೀವಲ್ಲ, ಅದೇ ರೀತಿಯಾಗಿ ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಫಾರ್ಮೇಷನ್ ಆಗಬೇಕಾದ್ರೆ ಅಲ್ಲಿ ಏನು ಮಾಡ್ತದೆ, ಸೆಲ್ಸ್ಗಳು ಒಂದಕ್ಕೊಂದು ಅಂಟಿಕೊಂಡು ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ ಇದ್ದು ಫಾರ್ಮೇಷನ್ ಆಗ್ತದೆ. ಸೊ ಅದಕ್ಕಾಗಿ ಸೆಲ್ಸ್ಗಳನ್ನು ನಾವೇನಂತ ಕರಿತೀವಪ್ಪ ಅಂದ್ರೆ ಸ್ಟ್ರಕ್ಚರಲ್ ಫಂಕ್ಷನಲ್ ಯೂನಿಟ್ ಆಫ್ ಲೈಫ್. ಅದು ಸ್ಟ್ರಕ್ಚರನ್ನು ಪ್ರೊವೈಡ್ ಮಾಡ್ತವೆ, ಒಂದು ಆರ್ಗ್ಯಾನಿಸಮ್ಗೆ ಪ್ರಾಪರ್ ಸ್ಟ್ರಕ್ಚರನ್ನು ಕೊಡ್ತವೆ, ಅದರ ಜೊತೆಗೆ ಫಂಕ್ಷನನ್ನು ಕೂಡ ಮಾಡ್ತವೆ. ದಟ್ಸ್ ವೈ ಸೆಲ್ ಇಸ್ ಕಾಲ್ಡ್ ದ ಸ್ಟ್ರಕ್ಚರಲ್ ಆ್ಯಂಡ್ ಫಂಕ್ಷನಲ್ ಯೂನಿಟ್ ಆಫ್ ದಿ ಲೈಫ್. ನೆಕ್ಸ್ಟ್, ವೈ ಇಸ್ ದ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಇಸ್ ಕಾಲ್ಡ್ ಸೆಲೆಕ್ಟಿವ್ಲಿ ಪರ್ಮಿಯಬಲ್ ಮೆಂಬ್ರೇನ್? ಈ permeable means what? Permeable means it can allow some material inside the cell. permeable ಅಂದ್ರೇನು? ಅಲೌ ಮಾಡುವುದು, ಒಳಗಡೆ ಬರಲಿಕ್ಕೆ ಅಲೌ ಮಾಡುವುದನ್ನು ನಾವು ಪರ್ಮಿಯೇಬಲ್ ಅಂತ ಕರಿತೀವಿ. ಸೆಲೆಕ್ಟಿವ್ಲಿ ಪರ್ಮಿಯೇಬಲ್. ಸೆಲೆಕ್ಟಿವ್ ಅಂದರೆ ಕೆಲವೊಂದು ಅಂಶಗಳನ್ನು ಮಾತ್ರ ಅದೇನು ಮಾಡ್ತೈತಿ ಪರ್ಮಿಷನನ್ನು ಕೊಡ್ತೈತಿ, ಕೆಲವೊಂದನ್ನು ಮಾತ್ರ. ಓಕೆ, ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಅಥವಾ ಸೆಲ್ ಮೆಂಬ್ರೇನ್ ಏನಂತ ಕರಿತೀವಪ್ಪ ಅಂತಂದ್ರೆ ಸೆಲೆಕ್ಟಿವ್ಲಿ ಪರ್ಮಿಯೇಬಲ್ ಮೆಂಬ್ರೇನ್ ಅಂತ ಕರಿತೀವಿ. ಯಾಕೆ ಅಂತ ಕೇಳ್ತಾರೆ. ದ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಈಸ್ ಅ ವೆರಿ ಡಿಸ್ಟಿಂಕ್ಟ್ ಸ್ಟ್ರಕ್ಚರ್ ಈಸ್ ಮೇಡ್ ವಿತ್ ಲಿಪಿಡ್. ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಎದರಿಂದ ಆಗಿರ್ತದ? ಸೆಲ್ ಮೆಂಬ್ರೇನು ಇಟ್ ಈಸ್ ಮೇಡ್ ವಿತ್ ಲಿಪಿಡ್ಸ್ಗಳಿಂದ ಆಗಿರ್ತದ. ಸೊ ಲಿಪಿಡ್ಸ್ಗಳಿಂದ ಆಗಿರುವುದರಿಂದ ಈ ಲಿಪಿಡ್ಸು ಮತ್ತು ಪ್ರೋಟೀನ್ಗಳಿಂದ ಆಗಿರುವುದರಿಂದ ದ್ಯಾಟ್ ಸೆಲೆಕ್ಟಿವ್ಲಿ ಅಲೌ ದ ಎಂಟ್ರಿ ಆಫ್ ಸಮ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್. ಇಟ್ ಅಲೌಸ್ ಓನ್ಲಿ ಸಮ್ ಮಾಲಿಕ್ಯೂಲ್ಸ್ಗಳನ್ನ ಮಾತ್ರ ಏನು ಮಾಡ್ತದೆ? ಅದು ಅಲೌ ಮಾಡ್ತದೆ. ದ್ಯಾಟ್ಸ್ ವೈ ಸೆಲ್ ವೈಲ್ ಪ್ರಿವೆಂಟಿಂಗ್ ಎಕ್ಸಿಸ್ಟ್ ದ ಅದರ್ಸ್ ಮೇಕಿಂಗ್ ದ ಸೆಲೆಕ್ಟಿವ್ಲಿ ಪರ್ಮಿಯಬಲ್ ಮೆಂಬ್ರೇನ್. ನೆಕ್ಸ್ಟ್ ಕಮ್ ಟು ನೆಕ್ಸ್ಟ್ ಕ್ವಶನ್. ಇಫ್ ದ ಆರ್ಗ್ಯಾನಿಸಮ್ ಆಫ್ ದಿ ಸೆಲ್ ಡಿಸ್ಟ್ರಾಯ್ಡ್ ಡ್ಯೂ ಟು ಸಮ್ ಫಿಸಿಕಲ್ ಆರ್ ಕೆಮಿಕಲ್ ಇನ್ಫ್ಲೂಯೆನ್ಸ್, ವಾಟ್ ವಿಲ್ ಹ್ಯಾಪನ್? Because all of the cell components are digested by its lysosome. Very important character this one. The organism of the cell is destroyed due to some physical or chemical. ಫಿಸಿಕಲ್ ಅಥವಾ ಕೆಮಿಕಲ್ದಿಂದ ಒಂದು ಸೆಲ್ ಇದ್ದು ಡ್ಯಾಮೇಜ್ ಆದರೆ ಏನಾಗ್ತದ ಅಂತ ಕೇಳ್ತಾ ಇದ್ದಾರೆ ಇಲ್ಲಿ ಕ್ವಶನ್. ಇಲ್ಲಿ ಆನ್ಸರ್ ಏನಾಗ್ತದೆ? ಇಲ್ಲಿ ಎವ್ರಿ ಕಂಪೋನೆಂಟ್ ಆಫ್ ದ ಸೆಲ್ ಈಸ್ ಕಂಟೇನ್ ಅ ಲೈಸೋಸೋಮ್. Every component has contained a lysosome. The lysosome if organism is damaged by some physical or a chemical action, the cell will not survive. Lysosome ಇದ್ದು ಏನು ಮಾಡ್ತದ? ಆ ಸೆಲ್ಲನ್ನು ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡ್ತದ. ಅದನ್ನು ಏನು ಮಾಡ್ತೈತಿ? ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡಿ ಅದನ್ನು ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡ್ತದ. Okay, come to why are lysosome known as suicide bags? Why are lysosome known as suicide bags? Lysosomes are the organelles inside the cell which contain hydrolytic enzymes. Lysosome, it contain hydrolytic enzyme. That is very important character of the lysosome. In that cell is injured, its lysosome may be blast and allow the enzyme to digest cell itself. As a result, it can call suicide bag of the cell. Why lysosome is called suicide bag of the cell? Because lysosome has contained a hydrolytic enzyme. ಕಿಣ್ವಗಳನ್ನು ಹೊಂದಿರ್ತಿದೆ. ಆ ಕಿಣ್ವಗಳು ಏನು ಮಾಡ್ತಾವೆ ಅಂದ್ರೆ ಬ್ಲಾಸ್ಟ್ ಆಗಿ ಹೊರಗಡೆ ಬಂದು ಅದೇನು ಡ್ಯಾಮೇಜ್ ಆದಂತಹ ಸೆಲ್ಸ್ಗಳನ್ನು ಏನು ಮಾಡ್ತಾವೆ? ತಿಂದು ಬಿಡ್ತವೆ. ಸೊ ಅದಕ್ಕೆ ನಾನು ಲೈಸೋಸೋಮ್ಗಳನ್ನ ಏನಂತ ಕರಿತೀವಿ? ಸುಸೈಡ್ ಬ್ಯಾಗ್ ಆಫ್ ದ ಸೆಲ್ ಅಂತ ಕರಿತಾ ಇದ್ದೀವಿ. ವಾಟ್ ವುಡ್ ಹ್ಯಾಪನ್ ಇಫ್ ದಿ ಪ್ಲಾಸ್ಮಾ ಮೆಂಬ್ರೇನ್ ಇಸ್ ರಪ್ಚರ್ ಆರ್ ಅ ಬ್ರೇಕ್ ಡೌನ್? That is important question. What would happen if a plasma membrane or a cell membrane is rupture or a breakdown? Here also same as it is answer. If plasma membrane or cell membrane is ruptures, the cell has been injured. In that case, the lysosome is active. Lysosome ಇದ್ದು ಆಕ್ಟಿವೇಟ್ ಆಗ್ತದೆ ಆ್ಯಂಡ್ ದೆನ್ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡಿಬಿಡ್ತದೆ. ಸೆಲ್ಸ್ಗಳನ್ನ ಡಿಸ್ಟ್ರಾಯ್ ಮಾಡಿ ಅದೇನು ಮಾಡ್ತದೆ? What would happen to the life of a cell if there is no Golgi apparatus? If Golgi apparatus are play very important role in cell, it help to preservation, modification and packaging and of the product in particles. All our task takes place by Golgi apparatus. All type of storage, modification, packaging and dispatch of the material within the cell, it would be possible because of the Golgi apparatus. That's why Golgi apparatus are very important. Okay, which organelle is known as the powerhouse of the cell? Mitochondria is we call it as a powerhouse of the cell. Mitochondria is really synthesis the energy in the form of ATP. ATP is adenosine triphosphate. Adenosine triphosphate. Adenosine triphosphate, ATP. During respiration process, energy is released in the mitochondria in the form of adenosine triphosphate during respiration process. Okay, That's why mitochondria is called powerhouse of the cell. Why do the lipids protein constituting the cell membrane get synthesized? There are two type of protein or two type of endoplasmic reticulums are present. Cell membrane gets synthesized. There are two types. One is smooth endoplasmic reticulum, another one is rough endoplasmic reticulum. Okay, that is smooth endoplasmic reticulum, SER, rough endoplasmic reticulum, RER. SER is a charge producing lipid that make up the cell membrane. SER produced lipids that help to cell membrane. RER is rough endoplasm. Why it is called rough endoplasmic reticulum? Because the endoplasmic reticulum is attached with ribosome. The surface is looking very rough. Why? Because ribosomes are attached to this endoplasmic reticulum. That's why it is called rough endoplasmic reticulum, which is responsible for production of protein that help to production of protein in the cell membrane. Okay, come to next question. How do substance like carbon dioxide and water moves in and out of the cell? Okay, here one thing you will understand here: carbon dioxide and oxygen, they are exchanged by diffusion method. What is method here? Diffusion is a process from molecules are moved to higher concentration level to lower concentration level. Molecules are moved from high to lower concentration. In that process, in that base, CO2 and O2 are takes place in our body. ನಮ್ಮ ದೇಹದಲ್ಲಿ ಕೂಡ ಏನು ಮಾಡ್ತಾವೆ? ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಇರ್ಬೋದು, ಆಮ್ಲಜನಕ ಇರಬಹುದು. ಹೈಯರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಡೆಯಿಂದ ಲೋವರ್ ಕಾನ್ಸಂಟ್ರೇಷನ್ ಕಡೆಗೆ ಮೂವ್ಮೆಂಟ್ ಆಗ್ತದ. ಇದರ ಮುಖಾಂತರ ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಮತ್ತು ವಾಟರ್ ಆಕ್ಸಿಜನ್ ಇದ್ದಿದ್ದು ಒಂದು ಕ್ಯಾಪಿಲರೀಸಿಂದ ಬ್ಲಡ್ ಕ್ಯಾಪಿಲರಿಯಿಂದ ಮತ್ತೊಂದು ಬ್ಲಡ್ ಕ್ಯಾಪಿಲರಿಗೆ ಪಾಸ್ ಆಗ್ತಾ ಆಗ್ತಾ ಹೋಗ್ತಿರ್ತಾವೆ. ಓಕೆ, ದಟ್ ಈಸ್ ದ ಪ್ರೋಸೆಸ್. ಕಮ್ ಟು ನೆಕ್ಸ್ಟ್. Okay, here prokaryotic cells, eukaryotic cells, some difference are there here. Okay, prokaryotics mean primitive organism, eukaryotic means new organism including human beings is also coming to eukaryotic organisms. Prokaryotes are a primitive organism. The size generally they are measured in micrometer, 1 to 10 micrometer. The eukaryotic cells are generally they are little bit larger in size. They are 5 to 100 micrometer. Nuclear region not defined, known as nucleoid, but in that eukaryote they are contain well defined surrounded nuclear membranes. Chromosomes are single stranded, but in a eukaryotic they are contain a more number of chromosomes, but chromosomes are double stranded in structure. Okay, membranes bound cell organelles are absent. Here membrane bound cells organelles are present. Okay, some difference are there here in a plant cell and animal cell. That is a cell wall is present, cell wall is absent, plastids are present, plastids are absent. Golgi bodies are present in different type, but in animal cell it is present well equipped. There are no centrosomes and centrioles, but centrosomes and centrioles play very important role in animal cells. The size of the vacuoles is bigger, the size of a vacuole is smaller. Vacuoles are present in both plant cell and animal cell, but vacuole size is differ in a plant and animal cell. Okay, see, this is the plant cell and animal cell. See the structure. The thick cell wall is present. Here also cell wall is present. Okay, large vacuole is present here, but here we can see it a small vacuoles. Centrioles are present here, centrioles are absent. Okay, same as it is prokaryotic cell and eukaryotic cell differences. nucleus is missing, nucleus is present, chromosomes are single stranded, chromosomes are several in number, organelles are attached with the cell membrane is absent, organelles are attached to the cell membrane. Okay, next question, how does amoeba obtain its own food? Amoeba feeds a planktonic bacteria. Amoeba, it feeds only planktonic bacteria which is float on surface of the water. It grows artificial feet, that is we call it as a pseudopodia, false foot. Pseudopodia, that is we call it as a false foot. False foot help to movement of the amoeba. Okay, it is a sac like structure is called food vacuole. Within the food vacuole digestion is occur. I'll show here structure here. Okay, see the structure, these are the amoeba structure. This is a food vacuole, small red structure, and inside the nucleus. Okay, and pseudopodia move towards the that food and capture the whole food and intake a food. Okay, what is osmosis? The passage of water through the semi permeable membrane from higher concentration. This is also osmosis and diffusion, both are same process, but difference is here diffusion is move from higher to lower without any membrane, but here osmosis higher level to lower layer with semi permeable membrane. Semi permeable membrane, that's why this is a difference. Okay, this is a difference, osmosis and higher. Okay, these are the some questions. ನಾನು ಇಲ್ಲಿ ಕೆಲವೊಂದು ಕ್ವಶನ್ಸ್ ಅನ್ನು ಇಲ್ಲಿ ತಂದಿದ್ದೀನಿ. ಐ ಹೋಪ್ ನಿಮ್ಗೆಲ್ಲರಿಗೂ ಅಂಡರ್ಸ್ಟ್ಯಾಂಡ್ ಆಗಿದೆ ಅಂತ ತಿಳ್ಕೊಂಡಿನಿ. ಏನಾದರೂ ಡೌಟ್ ಇದ್ದರೆ ಕಮೆಂಟ್ ಮಾಡಿ, ಲೈಕ್ ಮಾಡಿ, ಶೇರ್ ಮಾಡಿ, ಸಬ್ಸ್ಕ್ರೈಬ್ ಮಾಡಿ. ಓಕೆ, ಧನ್ಯವಾದಗಳು. ಥ್ಯಾಂಕ್ ಯು.